Okay, so this will be our last lecture together. We'll be going over Chapter 25, Reproduction and Development, and then next week will be your final. I'll post it on Tuesday and expect it turned into me by the end of that week. The due date will be posted with it, and I'll send an email to remind you. The learning objectives will be over sections 1, 8 through 9, and 12 through 16. We'll talk about reproductive technology and ethical issues related to that, the hormones involved in ovulation and menstruation. We'll discuss the process of fertilization, focus on embryonic development of the human, and pregnancy in general. We'll talk about how the embryo becomes male or female gender, and then we'll wrap up with hormones specific to childbirth and lactation. So reproductive options are on the rise, but in parallel to that, ethical issues are on the rise as well. The more we know about it and how it works, the more complicated it seems to become. You can probably think of a court case or an ethical dilemma or political issue that involves reproduction. So just think about some of those examples that you may come across in the media and how many of these issues have arisen relatively recently because new knowledge has allowed us to intervene in the reproductive process in some way. So this is just for you to think about. I can't take a poll online, but if a couple has frozen embryos but later divorce, who should retain custody of the embryos? So if you couldn't think of any examples of these ethical dilemmas that you've come across in the media, here's one for you right now. So technology is helping us a lot with conception, especially later in life fertility issues, but it's also giving rise to complex legal battles and ethical dilemmas that we've never encountered before. So let's talk about the hormones that orchestrate the process of ovulation. We'll be talking about the ovarian cycle, which is the timing and development of egg production in women. The ovarian cycle happens in parallel with the menstrual cycle, which is defined as the uterus preparing for egg implantation. And if an egg does not implant, then what's happening there is the shedding of the lining. That's the blood that occurs during menstruation. And both of these processes are regulated by hormones. Hormones regulate the timing and development of egg production, again called the ovarian cycle, which occurs approximately every 28 days. Hormones also regulate the menstrual cycle, during which the uterus prepares for the possible implantation and nurturing of a fertilized egg. If there is no fertilized egg that gets implanted, then the lining of the uterus is shed because fertilization didn't occur. So look at the menstrual cycle here. Menstruation occurs in the first 3 to 5 days. That's the bleeding that happens as the lining of the uterus is sloughed off. During that time, FSH, or follicle stimulating hormone, is produced. So as the uterine lining sheds, the levels of estrogen, which is the chief female sex hormone, they're going to drop, shown here. And as they drop, reduced levels of estrogen cause the pituitary gland to release follicle stimulating hormone, or FSH. FSH then causes a few follicles in the ovaries to grow and develop. So a follicle is where an egg is going to develop, essentially. So menstruation occurs, which means there was no egg. So this whole ovarian cycle is going to start again with the start of menstruation, which causes a decrease in estrogen. That decrease in estrogen causes that increase in FSH, and that increase in FSH causes that follicle to develop, to start making a new egg. As the follicle develops, it's going to produce estrogen. So we're going to see this gradual increase in the level of estrogen in the blood. The high levels of estrogen will then trigger the release of luteinizing hormone and more FSH. So you can see here at 14 days, there's this peak in the luteinizing hormone. The luteinizing hormone then triggers ovulation. So that's going to be day 14, ovulation, causing the egg to erupt from the follicle and out of the ovary. Okay, so the follicle is the cover where the egg is inside. That's being developed the first 14 days of this cycle. On day 14, because of that spike in luteinizing hormone, the egg releases from that follicle and it gets out of the ovary. That's why the time of ovulation, or day approximately 14 of your menstrual cycle, is the best time to get pregnant, because an egg has been released and it's available to get fertilized. When an egg is released, progesterone hormone is released. And what progesterone does is it causes the body to prepare for gestation of an embryo in case fertilization occurs. So here is where that lining in the uterus is going to start thickening again in response to that increased level of progesterone, saying, hey, let's make the lining in this uterus nice and thick to support implantation of a fertilized egg that we can then nourish for the first few days of development. And then, of course, the cycle will continue. If there is no fertilized egg that implants there, that lining is going to shed off in the beginning of the next cycle in the process of menstruation. So this is just everything I said written down for you, so you can re-reference this and go over it in your own time. It's a lot of information, but if you try and follow the steps of the hormones compared to the menstrual cycle, compared to the ovarian cycle, you can figure it all out. So the whole cycle starts with menstruation for 3 to 5 days of bleeding. That's the lining of the uterus getting pushed out because there was no fertilized egg. This is going to cause the decreased estrogen, which causes the increased FSH, which causes the follicle to develop with the egg inside. And then day 14, there's a bunch of estrogen produced that triggers luteinizing hormone. All of that is going to result in ovulation, meaning that the follicle erupts and the egg comes out of the ovary, which is the best time for fertilization of that egg, i.e. getting pregnant. When ovulation happens, the body wants to get ready to implant a fertilized egg, so progesterone is produced and the lining of the uterus, or the endometrium, thickens. And if there is no fertilized egg that implants, this whole cycle will start again with menstruation. The fate of an egg and the preparation of the endometrium, which is the inner lining of the uterus, can go in one of two directions depending on whether or not the egg is fertilized. You probably already picked up on this from the previous 3 slides, but I'll hit it again. So at this point, the process can go in one of two directions depending on whether or not the egg that was released during ovulation is fertilized. If the egg is not fertilized, shown here on the top, so there is no sperm reaching this egg, the egg will disintegrate and the corpus luteum, which is the surrounding of the egg, is going to shed. So everything disintegrates, the egg and everything else, which is going to remove the source of the hormones estrogen and progesterone, causing the lining of the uterus to slough off as menstrual blood. With continuing reduction in estrogen levels, the pituitary gland is spurred to release follicle stimulate, the FSH, follicle stimulating hormone, again to restart that whole process of making an egg and ovulation and all that for the next month. Here on the bottom, if the egg is fertilized because the sperm reached the egg, this usually occurs in the fallopian tube. Then the zygote, which is the fertilized egg, will begin to develop, and after several days it will start secreting its own hormone known as HCG, which is human chorionic gonadotropin. You don't need to know that name. But this hormone prevents degradation of the corpus luteum, which is the surrounding of the egg. And if the corpus luteum is kept, then progesterone will be secreted, which will maintain that thick inner lining of the uterus instead of it getting sloughed off. I just want to point out here with all of these hormones, menopause is a cessation, meaning the end of ovulation and menstruation for women. It usually occurs between the ages of 45 and 55. It's not clear whether this is an evolutionary adaptation. Menopause, most species other than humans, females remain fertile throughout their entire adult life. So why don't we as well? It may be a consequence of the fact that humans live for a really long time compared to any other species. At some point, we talked about this with Down syndrome, at some point the eggs are going to get older. As the eggs are getting too old, you can see chromosomal abnormalities happen, and then you can get children with disorders like Down syndrome due to those chromosomal abnormalities. So that's most likely why women are not able to reproduce throughout their later lives. Alright, so you probably need to pause this and go back and figure out which one it is. But which hormone is involved in preparing the uterus for gestation of an embryo? Gestation just means it's going to implant and get nourished. Right, the answer is number 4. So when progesterone levels are high, that's when we're going to see that thickening of the lining of the uterus. If progesterone levels are low, that's when we're going to see that sloughing off of that lining as menstrual blood. In the ovaries, a cell within a follicle is stimulated to develop into a fertile egg by secretion of hormones estrogen, FSH, and luteinizing hormone. So you should know those 3. The preparation of the uterus for implantation of a fertilized egg is coordinated by the hormone progesterone. You should know that. Okay, in fertilization, 2 cells become 1. So an egg cell was made through meiosis, so it is haploid. That means that it has 23 chromosomes, 1 of each. The sperm cell also produced by meiosis, which means it's haploid, it has 23. So when they come together, you will have a complete set of 46 chromosomes. The process of fertilization is actually pretty cool. I posted some videos about this months ago, but they are on the YouTube channel. The process of fertilization isn't as simple as sperm meets egg. Penetration is necessary and involves multiple steps that are designed to prevent the entry by multiple sperm into the egg. So one of the questions I get asked a lot is, well, how do twins happen? You know, is it 2 sperm are going to attach to the same egg? No, your body naturally blocks a second sperm from entering, because think about it, you would then have 23 too many chromosomes. Right? So you only want 1 sperm to fertilize the egg, but there are millions of sperm running around. So as soon as 1 sperm gets in there, the egg goes into lockdown mode to block any other sperm from penetrating it. So I'm going to talk about the details of that now. So after ovulation, the egg is still surrounded by some smaller cells. Those cells stand between the sperm and the egg as a barrier. There's also a thick protein layer called the zona pellucida. As sperm approaches the egg, there's a release of calcium by the cells surrounding the egg that helps to increase the sperm's swimming speed and tail movements in a process known as sperm activation. So as sperm get close, the egg is going to secrete calcium, which then makes the sperm swim even faster to try and get there. A second phase of sperm activation occurs as a sperm cell pushes its way through the barrier, and the head of the sperm dissolves and releases digestive enzymes that helps the sperm penetrate that zona pellucida. Once a sperm cell gets through that zone, the egg is activated and the plasma membrane of the egg and sperm fuse together. This activation and fusion has a couple of important consequences. First, it changes the egg's membrane in such a way that it is impossible for any other sperm to also fertilize the egg. Again, after all, it would be a genetic disaster for the fertilized egg to have more than 2 sets of chromosomes. Second, fusion of the plasma membranes induces the egg to complete the second meiotic division. On doing so, one haploid egg is formed along with a smaller polar body. That polar body then disintegrates or is ejected from the egg. You don't need to know that part, but definitely know that once a sperm gets in there and those membranes fuse, that the egg goes into lockdown mode. And finally, the haploid nucleus of the egg fuses with the haploid nucleus of the sperm, creating a diploid cell called the zygote. So you should know by now from all those previous chapters that a haploid sperm and a haploid egg get together to make a diploid zygote. So once the sperm penetrates that protective zone, the egg blocks additional sperm entry. And the fusion of sperm and egg result in a diploid zygote. This is a picture of a developing human fetus 5 months after fertilization. I also have some videos that I put up a long time ago as well on the YouTube channel regarding the 9-month development cycle of humans. Very interesting stuff. Early embryonic development occurs in these stages we call cleavage, gastrulation, and neurulation. Cleavage is early cell division. So you start with one cell. All humans just started as one cell zygotes. So early cell division happens from that one cell to become a ball of cells. And then we have gastrulation, which is where we start forming 3 distinct layers of tissue. And then neurulation is the formation of early organ systems from those 3 layers. So cleavage is where that single cell divides by mitosis. So now we're talking about mitosis, not meiosis. So mitosis, to form a hollow ball of approximately 1,000 cells. So it takes about 30 hours for that single cell that has a diploid genome to become 2 cells, and then it divides again 30 hours later, so it would become 4, and on and on and on. The cells divide continuously, forming this hollow ball filled with fluid, and then there's this mass of cells on top of that fluid. This is called a blastula at about a week after fertilization. Just want to point out that the overall size of the embryo remains the same in the blastula. The size of the cells just kept getting smaller and smaller. And that blastula, sometimes it'll be called a blastocyst, is made of approximately 1,000 cells. At the blastocyst stage, it grows rapidly and forms membranes that protect it. This includes the amniotic membrane, which surrounds the embryo. And we're also going to see the formation of the placenta, which is the structure that connects the developing embryo to the wall of the uterus. The placenta is packed with blood vessels that bring nourishment to the embryo and remove wastes generated by the embryo. So if you have a fertilized egg, your body is going to keep those progesterone levels high to keep the lining of the uterus thick so that you can have attachment of that blastocyst and the development of the placenta start happening. But the human embryo, after a week, once it's formed, this blastocyst stage is really small. You can see how small it is here in reference to the tip of a pen. The second phase of development is called gastrulation. This is where we see 3 distinct germ layers of tissue form. Picture a beach ball, a hollow sphere. Now imagine making a fist and slowly pushing inward on the beach ball. That resembles the process of gastrulation. The entire mass of cells is called a gastrula. The indentation is called the blastopore. So where your fist was punching in, that would be the blastopore. The blastopore is located almost opposite the point at which the sperm entered the egg originally. And in vertebrates, it becomes the anus. 3 distinct layers of tissue form during gastrulation. The cells in these layers have not yet been differentiated though, but they have become determined, which means that the type of tissue they will become is irreversibly decided at this point. Prior to determination, cells are referred to as totipotent. So you may have heard totipotent or pluripotent stem cells in stem cell research. Totipotent stem cells have the capacity to develop into any type of tissue in the body. Here, during gastrulation, cells from each of the layers are pluripotent. They become a lot of different things within that layer, but they're not totipotent anymore. So a cell from the mesoderm could become muscles or skeleton, but it could never become a hair cell that you would normally find from the ectoderm. So here's a summary of what happens. First, the blastocyst begins to fold inward, forming an indentation called the blastospore. As the cells continue to push inward, they are called endoderm, and they form the lining of what will eventually become the digestive tract. Endoderm cells will also form the liver, pancreas, lining of the respiratory tract. At this point, the entire mass of cells is called a gastrula. The cells that remain on the outer surface of the gastrula are called ectoderm and will ultimately form the outer layer of skin, the hair, the lining of the nose and mouth, and our nervous system. Some additional cells migrate inward between the endoderm and ectoderm, forming a third type of tissue called mesoderm, which gives rise to the muscles and skeleton. The specific details of gastrulation differ slightly from one species to the next. The general outcome, however, is the same throughout vertebrates. So that was week 2 of embryonic development. So now during week 3 of embryonic development, the 3 types of tissues formed during gastrulation begin to develop into the various organs and tissues. At this time, within the mesoderm and running the length of the embryo just above the digestive tract, we're going to see the notochord form. We talked about notochords before when we talked about chordates. The notochord is a flexible rod that develops in all chordates and gives support to surrounding tissues. In vertebrates, the notochord's function is ultimately taken over by the backbone. So that's going to become our backbone in humans or any vertebrates. Just above the notochord, ectoderm folds in for the entire length of the embryo, first forming a groove and then becoming a long hollow tube called the neural tube. The process is referred to as neurulation, and the resulting neural tube ultimately develops into the entire nervous system, including the brain and spinal cord. As the neural tube is developing, blocks of mesoderm tissue called somites begin to form down the length of the embryo. These somites will form the vertebrae and the muscles. Next to the somites, spaces form that will become the body cavity, within which many of the organs will form. At this point, toward the end of the 3rd week of development, the human embryo is only 2 millimeters, which is less than 1/10 of an inch, long. Further development and specialization occurs as some cells are programmed to die, in essence sculpting the body. Okay, so which step during embryonic development does notochord develop? Pause if you need to. The answer is, of course, neurulation. So those are the 3 steps of early development in the first 3 weeks: cleavage in the first 6 days, then gastrulation to form those 3 layers, and then neurulation in the 3rd week. For the sake of time, we're going to skip some of the development cycle, but we will talk about gender determination. By gender, I mean male or female. And you should remember the sex chromosomes and which combination of sex chromosomes equal male and which combination equal female. But to jog your memory, try and answer this question. XX is female, so male is XY. This is a busy slide, but you should take some time, pause the video, and go over it. During the first few weeks after fertilization, surprisingly little distinguishes males from females. About 4 weeks into embryo development in mammals, there's a gene carried on the Y chromosome called the SRY gene SRY stands for sex determining region on the Y chromosome, and it can initiate development as a male or female. The gene is expressed in the tissues that will form the gonads, and usually when SRY is expressed, the gonads develop as testes. If SRY is not present, then the ovaries develop instead. So this process starts about 4 weeks into development. Male and female don't really look different. The default is to become female, actually, unless that SRY gene is present because you have a Y chromosome around, which is only found in males, and then you'll get the development of the testes. This seems really straightforward, and it's always been presented as very straightforward, the development of male and female, but we know it's not that straightforward. At several points in development, the development of an embryo as male or female can be disrupted. The SRY gene could be moved to the X chromosome instead of the Y, so even if an individual is XX, they could have one of their X chromosomes expressing that SRY gene, and then they would have incomplete development of female gonads. There's other things called androgen insensitivity syndrome, which is where body tissues are unresponsive to testosterone's effects. Okay, so the SRY gene, when it's present, is going to trigger the development of testes. How does it do it? It's because it's going to change the testosterone levels. So even if an SRY gene is expressed and turning on a high level of testosterone, if the cells in the developing male's body aren't responding to that testosterone, then the body is going to develop as female even though there's an XY sex chromosome combination. Another example would be 5-alpha reductase deficiency. This would be with XY individuals carrying a non-functioning version of the gene that converts testosterone into DHT. Alright, so let's go back over here. So the SRY gene is present on the Y chromosome. It's going to turn on and determine testosterone levels, which are going to change the internal sex structures toward male. And then that testosterone is going to affect DHT, which is going to trigger the external sex structures as male. So if you have 5-alpha reductase deficiency, the testosterone is going to get converted into DHT, resulting in individuals that have testes, but externally they're not having male reproductive characteristics, so the body will develop as female. And then at puberty, high levels of testosterone from the testes inside will cause development of male secondary sex characteristics and external genitals. So this wouldn't even be something that would start affecting this individual's gender until they hit puberty. I want to tell you about that you don't need to know, but just so you're an informed citizen. There's a lot of disorders. There's congenital adrenal hyperplasia, in which overproduction of hormones in the adrenal gland causes masculization of the genitals in female infants. I already talked about 5-alpha reductase deficiency, which causes incomplete masculization of the genitals in male infants. Partial androgen insensitivity that we talked about, in which cells do not respond to testosterone, causing incomplete masculinization of the genitals in male infants. There's penile agenesis, in which male infants are born without a penis, so that would go under the ambiguous genitals category. There's differing levels of that androgen insensitivity. There's also Klinefelter syndrome and other syndromes like Turner syndrome, in which you don't have the correct combination of sex chromosomes. So XX should equal female and XY should equal male. But what happens if you have an extra X around, or you're missing the Y chromosome because of those chromosomal abnormalities we talked about, similar to Down syndrome, except instead of this happening in chromosome 21 like in Down syndrome, this is happening in the sex chromosomes. So in Klinefelter syndrome, that's when you have a male infant born with an extra X chromosome, which is the female chromosome, which will cause incomplete masculization and other anomalies. Turner syndrome is when female infants are born with one rather than two X chromosomes, causing developmental abnormalities. Let's go 3 stages of pregnancy quickly. The development of a human embryo and fetus is usually developed into 3 equal periods of approximately 3 months each. The fetus stage begins about 8 weeks after fertilization. So when somebody says fetus, they don't mean the fertilized egg, the zygote. They're not talking about 8 weeks after fertilization. That's defined as when the organs and other major structures first form. Each of these trimesters is characterized by specific features and developmental milestones. So we have the first trimester where cells begin to differentiate, obviously. So within the first 3 weeks, we're going to have that neurulation process occurring to form the notochord. And then after that, for the next 3 months, we'll see the forming of major organs and structures begin. At 8 weeks, we will have a fetus with organs and other structures forming. Second trimester is significant muscle and bone growth, but we don't see a lot of new development of organs or anything like that. That mostly happened in the first trimester. And the third trimester is where significant development of the nervous system takes place. So let's look at the first month. In the first month, within the first 30 hours, that zygote, which is the fertilized single cell, is going to have the first cleavage occur, the first cell division. Within 60 hours, the second cleavage occurs, and there's no overall growth, just cell division. Within 6 to 7 days, the embryo reaches the uterus for implantation in the endometrium. The endometrium is that thick lining within the uterus that if we don't have an egg implant there, that's why you see menstruation happen. And that's why when women are pregnant, they don't menstruate, because you want to keep that lining around to help nourish the baby. Within the second week, gastrulation occurs. The placenta also forms in that second week. It forms as 2 membranes growing around the blastocyst. The placenta is defined as a massive tissue in which nutrients, respiratory gases, and waste products are transferred between the circulatory systems of the mother and the developing embryo. The embryo is connected to it by the body stalk, which will become the umbilical cord. In the third week, neurulation occurs, and then the fourth week, major structures and organs, including the eyes and heart, begin to form. At this point, it is also possible to see the arm and leg buds. So at the end of the first month, this is what the human will look like. You see the placenta is here within the lining of the uterus. The embryo is connected through the umbilical cord, and there's these little buds that will eventually become the arms and legs. During the second trimester, the skeletal and muscular systems develop rapidly, and the mother begins feeling the kicks and movements of the baby. So that's why babies start kicking in the second trimester. The heartbeat becomes much stronger and then can be easily heard with a stethoscope. So that's why you go in to the doctor when you go in to check on your baby's heartbeat. And the overall size of the fetus increases about 1.3 pounds and 12 inches. So at the end of the first trimester, the baby is a few ounces, and it's about 2 or 3 inches long. That's it. At the end of the second trimester, the baby's going to be about 1.3 pounds and 12 inches long. Then the final 3 months sees a rapid growth and development of the nervous system and a dramatic increase in the weight and size. The fetus continues to increase size and weight until 40 weeks. A healthy weight is between 5 pounds 8 ounces and 8 pounds 8 ounces. The minimum weight needed for survival is only 1.5 pounds though. This whole process is of course 9 months of development in humans. I'm going to briefly talk about some of the hormones in childbirth and then lactation. In a process that's still not fully understood, the fully developed fetus produces hormones that cause the placenta to release hormones, including estrogen and prostaglandins. Those hormones being released by the placenta are going to stimulate contractions of the uterus. This hormonal surge can be artificially replicated to induce these contractions. So when they say they're going to induce labor, that would be that they are injecting a surge of these hormones because the baby and or the placenta aren't secreting them themselves to start those contractions. The contractions increase in frequency and intensity as birth nears. The opening of the cervix dilates. So when they talk about the dilating cervix, it's dilating from 3 centimeters to 10 centimeters or more. Upon reaching 10 centimeters of dilation, the mother is instructed to begin pushing to deliver the baby, which through the vagina, which now functions as a birth canal. The baby usually emerges head first. There's then a brief relaxation of the contractions before the third and final phase of the birth process. That is when contractions resume to get rid of the placenta, and the placenta is sheared from the walls of the uterus and expelled. So this whole process of birth is a huge surge of hormones, like very, very high levels of hormones happening to induce those contractions. So you've probably heard of postpartum depression. You may have even heard of the baby blues. This is common, especially the baby blues. 70 to 80% of all new mothers experience some sort of negative feeling or mood swings after the birth of their child, and that's because they're coming off of this dramatic high of hormones. So this is the baby blues, but a lot of women have felt stigmatized to not talk about it or bring it up at all, and now that it's becoming a little less stigmatized, we can see how common it is. And even something more dramatic like postpartum depression occurs in approximately 15% of new mothers. Again, this is because hormone changes occur during pregnancy and then again after the baby is born, may produce chemical changes in the brain resulting in depression symptoms. So that doesn't mean somebody is a bad mother or they're not fit to have their child. They're just going through some hormone changes because of that child process. There are hormones that then stimulate lactation. During pregnancy, mammary gland development is stimulated by estrogen, progesterone, and other hormones to prepare the mother to nourish the baby after birth. After birth, the release of prolactin stimulates milk production, and suckling or crying by the baby stimulates oxytocin production. Oxytocin production is important because it stimulates milk ejection from the breasts. For the first few days, a yellow substance high in proteins and antibodies is produced, which is gradually replaced by milk that is higher in fats and sugar. So the breast milk does change in its look and its composition. During lactation, the suckling of the infant prevents the pituitary from releasing a surge of luteinizing hormone to cause ovulation. For this reason, during lactation, a woman's fertility is significantly reduced, but this is not like a foolproof child control method. I've known women that breastfed and still got pregnant. And that's because there's several conditions that must be met for the woman's fertility to be reduced by breastfeeding. The mother must be breastfeeding the child exclusively, and also the baby must be less than 6 months old, and no more than 6 hours can pass between any two feedings. So, if you're not watching those 6 hours carefully, then this is not a really reliable form of birth control. More about oxytocin. It's a hormone produced by the pituitary gland in the brain. In addition to stimulating release of the milk through the breast, it's also going to increase relaxation. It lowers stress and anxiety. It lowers blood pressure. It causes muscle contractions for childbirth too. It's the hormone involved in social relationships like bonding, trust, and love. So not just bonding between a mother and her baby, but any sort of bonding, loving relationship is going to have oxytocin hormones involved. So oxytocin is an important hormone for women during childbirth, causing the uterus to contract and push out the baby, important for lactation, and important for that bonding process between a mother and her infant during breastfeeding. Hormones play a major role in reproduction. Which hormone is responsible for both lactation and trust? Pause the video if you need to. The answer is, of course, oxytocin. Okay, so we talked about the birth process. There's 3 different phases of it. The first was contraction and dilation. The second was delivery of the baby. And the third was expulsion of the placenta. We then talked about lactation and the hormone oxytocin. We'll end by talking about reproductive technologies just briefly. Here we're showing the DNA from a single sperm being injected into the cytoplasm of an egg. Technological advances have led the development of techniques for successful reproduction in situations where couples previously would have remained infertile. It has also expanded the childbearing years for women. This can become important for women with careers that wait to have children until later in life than they used to. Infertility is defined as the inability to get pregnant after 1 year of trying. It affects approximately 7% of married couples in the United States in any given year. There are many causes of infertility, evenly divided between males and females, by the way. It can include low sperm counts and insufficient sperm motility, meaning that you have enough sperm, but they're not swimming fast enough to get to the egg. There could also be ovulation problems or issues with the fallopian tubes. The fallopian tube is the part in the female where fertilization takes place. And even in 10% of cases, no actual cause can be found for the infertility. So that's here at the bottom. That's when the eggs and sperm are put into the fallopian tube. They're not and they should hopefully fertilize there instead of having them fertilize in a petri dish. Okay, so that's it for assisted reproductive technology, or ART. That is also the end of the lecture.